Hello. Good evening, everyone. Hello. Mm, Good evening, everyone. Welcome to Big Six of online discussion wastewater treatment and recycling. The instructor of this course is Professor Manoj Kumar Tiwari. My name is Maniatnam Prakash, and I am a PhD scholar in Department of Civil Engineering at IIT Kanpur. So, as usual, we will be meeting on every Tuesday from 7 p.m. to 9 p.m. using this Google Meet, the link for which has been already shared with you. And the recording of this session is being uploaded on YouTube channel, that the link for which has been already shared with you. So, in week 6, we have gone through these four different topics. So, if you recall, our previous discussion was about primary sedimentation tank. So, that was primary treatment. Okay, now so we know that there are different mode of treatment. That actually I have one slide recapping all those different unit of treatment. But in this week 6, we have mainly focused on biological treatment of wastewater. So we know that a huge amount of organic matter is present in our wastewater, and that has to be removed before disposal or before reuse. Okay, so and we know that for degradation of organic matter there is a need of biological the organic matter can be degraded by the means of some biological means, so that with the help of microbes. So how those microbes, how their life vary with how the microbes behave in presence of organic matter, how this treatment happen, what are the different uh, um, factor which is influencing our biological treatment, we will be looking in this week. Okay, now so we have introduction to biological treatment of wastewater, then biological treatment of wastewater, microbial growth and its kinetics, how microbe grow and how its kinetics happen, at what rate. We have seen in we have I don't remember that we have discussed about chemical kinetics, but I have referred one book where they have discussed about this chemical kinetics. Okay, now so then we have biological treatment of wastewater. We have mainly focused on activated sludge process, and then other treatment plant like trickling filter, rotating circular batch. I don't remember the full form of RCB, which I will be looking in upcoming slide. Okay, now so this picture we have seen in last lecture, that any wastewater treatment have four unit, that is preliminary treatment, primary treatment, secondary treatment, and advanced treatment. Just wait for 1 minute, I'm coming. Sorry. So we have seen that there is preliminary treatment to remove boulders, yeah, grit and all these things from our wastewater. Then we have a screening, grit removal. Then we have primary treatment. We have seen in last class we have discussed about primary sedimentation tank, how there is concept of discrete particle settling. Then we have seen the terminal velocity or settling velocity, a scouring velocity, and different parameter to design this primary sedimentation tank. Then we have secondary tank. Yeah, secondary means biological also, like to remove the this unit only focus on removal of organic matter. So After primary sedimentation tank, the the wastewater come that whatever treatment has happened, that was for our suspended particles. Okay na, we have removed suspended particle, then we have add coagulants and flocculants and all these thing. Okay, then we, our main focus on for the treatment of wastewater is for organic matter present in our system. Okay na, so for that we have this thing, secondary treatment, and here we use some say the RBC full from by the rotating contactors. Okay na, we will see the picture how. And after secondary treatment means there is some advanced, if we want to reuse, yeah, dispose, depending upon that we have this advanced treatment unit. Okay na, so before going to the question and answer, I just want to discuss some of the concepts. That is, if you look carefully into a biological activated sludge process or biological sludge process. So we know that suppose if in my system there is the organic matter, and if we somehow introduce some microbes, then those microbes in presence of oxygen or in absence of oxygen, depending on aerobic condition or anaerobic condition, that microbe will degrade our organic matter. So okay now, so it has been observed that suppose there is one single unit, like in sedimentation tank, what we have seen that water was coming and it was settling on its own. So this so right now, similar, if we apply same concept in for our secondary treatment also, that there is a tank, wastewater containing organic matter will be there, and if we allow themselves to decompose on its own, because we know that there will be some microbes in my bacteria, or mean in my wastewater also. So it will take some time for it will take some time to decompose. So what we do to accelerate the process, what we do that we let it pass through some, we call it we co- so we, what happened that mhm, we introduce some of the some bacteria from outside. Okay, so there is a tank beside this chamber, and this contains some microbes, already activated microbes, because we will be seeing that there are different phase of microbes. Okay, now first they grow, then they have their stationary phase, lag phase, and all these things. So there is something called activated, means the microbes are fully activated. So instead of allowing them to decompose on its own, what we do that we recirculate some of the bacteria from nearby tank to our main tank, that contains our wastewater. So what will happen that it will it will activate the process of decomposition. That's why this whole thing is called activated sludge process, means we are producing sludge by means of we are activating it. How we are activating it? We are introducing some of the bacteria, microbes, which was in this tank to our treatment unit. Okay now, so here x is you can see that we are the x is biomass concentration and s is substrate concentration, and we are using the use of active microorganism to decompose the organic matter. So we are using active microorganism, which can degrade our which can degrade our organic matter in a faster way. That's why the term is known as activated sludge process. Okay now, the first thing which is required is oxygen requirement. Okay now, so depending upon if oxygen is available, which is mostly not available in our case. Okay now, because we know that a huge amount of organic matter is there, and all those organic matter will be decomposed. All these organic matter will consume our oxygen for their degradation. So organic if oxygen is present, we call that as aerobic condition. And if oxygen is not present, yeah, free oxygen is not present, then we say that this is an anaerobic oxygen. Okay now, so generally in our activated sludge process, this anaerobic condition prevails. Okay now, because there is lack of oxygen and all these things. Okay now, so organic carbon compounds consumed by microorganism and converted to carbon dioxide and biomass. Okay, we can see that converted to carbon dioxide, CO2, that is known as catabolism, and some new cells are also formed, so that is known as anabolism. Part of this biomass is oxidized by microorganisms to carbon dioxide. Energy produced through oxidation of organic matter to carbon dioxide is driving force for subsequent of the microorganism production of new biomass. Oxygen is essential for as terminal. So this is like why oxygen is required and all this thing. Okay, now you can see that this is our organic matter, and this O2 will be required in presence of acidic medium. CO2 and ammonium ions will present, and some energy will be released, and this energy will be again used for uh, again this energy will be used for what do we call generation of new cell and generation of CO2 and all this thing. Okay, now so we have different units in our activated sludge process. So mixing and aeration of the mixed liquor, wastewater mixed with activated sludge undergoes continuous mixing and aeration in aeration tank. This promotes the growth and activity of aerobic microbes which consume organic matter pollutant in the wastewater. So there is continuous supply of air. Okay, now so we don't want anaerobic condition. Okay, now because in an aerobic condition there will be no that there will be too much smell and all these things, some foul gas will be releasing and all these things. Okay, so we don't want that. So this promotes the growth and activity of anaerobic organism which consume organic pollutant present in the wastewater. Settling of the mixed liquor to the separate the sludge. After aeration, the mixed liquor is allowed to settle in the settling chain, settling tank or clarifier. During settling, heavy particles including activated sludge flocks settle to the bottom as sludge while the clear water rises to the top. Anoxic condition for D. So we know that we have both thing nitrogen and carbon. So carbon we know that it will be released in the form of some gas or some other form. Okay, now here you can see that this carbon was converted into what happened to this what happened to this nitrogen. For that we have this denitrification thing. In some ASP configuration, anoxic condition absence of free oxygen are created within the treatment unit to facilitate the denitrification, where NO3 is converted to N2 gas or nitrogen dioxide NOx by denitrifying bacteria. This helps in reducing the nitrogen content in the treated wastewater. Then we have flocculation thing. Flocculation involves the addition of some chemicals to promote the aggregation of one particle suspended in the wastewater into large. This flux settle rapidly during the settling removal of suspended. So in this thing in our biological treatment, because we know that some of there will be. So this you can relate with the sedimentation tank also. Okay, now same concept has been used here. Okay, so coming to the microbial growth, so we know that every microbes will will do some activity and finally it will die. Okay, now so what happened that when in an or in an organic system, if a system containing organic matter, if some microbes are introduced to decompose, so it goes to a different phase. The first phase is known as lag phase. So as soon as microbes are introduced in this organic matter, so first these microbes will start to adjust himself into the given condition depending on the pH, depending on the chemical composition of organic matter. It will start to adjust himself. So that is known as lag phase. Then we have exponential growth phase. So what will happen that in exponential growth phase, so at the microbes have adjusted himself, so what will happen then, rate of reproduction will increase because they are now, imagine two things. What is happening that the amount of food which is available for them is very high. So what will happen that they will start consuming this organic organic matter. Okay, na so as they have started to consume this organic matter, so what will happen that they are getting, we know that they are using this organic matter for to generate energy, and that energy can be used for generation of biomass. So what is happening that as huge amount of food is available, so what will happen that they will start to grow, their reproduction will start to increase, population will start to increase, and that is known as exponential growth phase. But after some time what will happen is that the food amount which was in huge amount has become somehow limited. So and at the same time microbes are also started to die. Okay now, so at the rate at which they are increasing their population, at the same rate they are dying also. That's why a phase come a stationary phase. And after some time what is happening that the amount of food which is available are not sufficient to fulfill the whole population, so what will happen that they will start dying. So that is known as declining growth phase. And at the end we have this is endogenous phase, because all the food all the food which was there, just 1 minute. Hmm. All the food which was there has finished. The number of bacteria has also finished. So that's why this phase, endogenous phase. The name itself suggests that it has declined. It has extinct. Okay now. The exponential growth, also known as the log phase. Remember, exponential log and lag both are different. Lag phase is just I'm not reading about lag phase. Lag phase is our just adjustment phase, that he the bacteria is just adjusting himself to the given environmental condition. Also known, this is period of intense growth and where population of microbes rapidly increase. During this phase, the rapid growth is at maximum as the microbes are actively dividing and replicating in favorable with abundant nutrients. Means huge amounts of foods are there. Okay, now so that's why they are growing too much. Okay, then we have a stationary phase. In this phase, the growth rate stabilizes, the number of viable cells remaining relatively constant. The occurs because the rate of cell division equals the rate of means death and division are equal. That's why the population is not increasing. It's typically characterized by plateau in the growth curve. Nutrients depletion, waste accumulation, and other environmental factors contribute to the selection of exponential growth and the entry into the stationary phase. Okay, now declining growth phase we have. After exponential growth phase, the growth rate begins to slow down due to depletion of nutrition. Means food which was there has finished. Okay, the accumulation of waste, huge amount of waste, because what will happen that all the microbes are dying, so huge amount of waste has been also generated. Okay, now in this phase, the growth rate increases and the number of viable cells may start to decline. However, it is important to note that all the cells may be dying at this stage, but the overall growth rate is diminishing. Means few cells are means most of the mm, cells are dying also. That's why there is a decrease in population. Okay. Then at the last we have endogenous phase. This is a phase where the number of viable cells start to decline and the cell enter a state of dormancy or undergo program dead cell. This phase is characterized by decrease in cell viability due to various factors such as nutrients exhaustion, toxic metabolic accumulation, and environmental stress. Okay. Eventually, the population may reach a point where cells are no longer viable, leading to the decline of the culture. Okay. So this was all about this our ASP, how these microbes and things occur. Okay, now then we have this thing, other type of bioreactors other than ASPs. Remember, this ASP is a kind of bioreactor. Okay, now so we have, and here we can see that this was our organic matter. Here there was microbes were suspended in the system. Okay, now but we have another thing that is known as trickling filter. Okay, now trickling filter, what will happen that a system will be there. It will contain some membrane. Okay, now and membrane will contain some microbes on them. Okay, some microbes will be attached to this membrane and wastewater will be introduced here. So what will happen that as soon as this wastewater come contact with this membrane containing some microbes, they will start to decompose the organic matter. So okay now, so here you can see it is a attached growth trickling filter. We have this thing rotating biological contractor. Here also we have same thing. There will be huge biological unit will be there, and what will happen that microbes will be attached to this rotator, and what will happen that as soon as the organic matter come in the contact of this rotator, it will start decomposing our microbes, organic matter. Okay now, then we have sequential batch reactors. This is suspended again, similar to this activator. This is suspended growth. Then we have mixed biofilm, hybrid suspended, both things suspended and hybrid are. Then we have membrane. Again here we have suspended. Okay now. So here you can see out of all these things I have discussed this sequential batch reactor. Like here you can see, remember sequential batch reactor. Sequential means one by one, and batch means no that a single means there is no flow of water. So what will happen that first wastewater will be filled, then what will happen that uh, it will pass through a definite amount of time will be given through each chamber. Okay now. So what will happen that it will be first filled, then there will be some reaction, and after some reaction means decomposition, then it will be settled, or whatever the organic matter which was produced will be settled down, and then what will happen that the clear water will be drawn out, and the sludge will be passed, and similarly this will keep going. So we can see that each thing is happening in a particular unit, so that's why it is known as sequential batch reactor. Suspended growth, sequential batch, fine bubble aeration in high quality element, moving system, buyers, and complex operation requiring electronics control. So here you can see this is a picture of this sequential batch reactor. First, I don't remember where water is coming, but I think preferably water is coming from this side. Okay now. Good evening, Rupali.
Good evening, sir.
So I was discussing about this biological treatment unit. So just join. I'm just giving you about this. Sorry, brief idea about different biological reactor. Okay, now so you can, if you have any kind of doubt, you can ask by raising your hand. Yeah, just by unmuting yourself. Then we have then with this thing we have a rotating biological contractor. This is kind of a attached bioreactor. Okay now, so you can see these are rotating films rotating, and these here is the real picture. Okay now, so and this here you can see miss it's not visual in this picture, but these biological things will contain some microbes, and what will happen that a inflow will come containing organic matter, and it will pass through different unit in this is a real picture from, I don't know, I have taken from Google. So what will happen that organic matter will pass through each and every biological rotating contractor. Okay now, so what will happen that as soon as they will come contact with the organic matter, the microbes will degrade our organic matter, and finally we will have, at the end of outflow, we will have this wastewater free of organic matter. Okay. Then last thing, we were discussing one thing that everywhere we were saying that organic matter will be decomposed by microorganism. So but the question is which microorganism, what kind of microorganism will decompose. So there is a like, this is like very in itself is a big topic, like which kind of microbes will decompose which type of organic matter. But I have prepared some slides where I will be showing that uh, which type of organic matters will be decomposed by which kind of bacteria and all these things. So it's not complete, but whatever we have, whatever was required for our discussion, I have included this thing. Okay now. So remember that we all are microorganism. If someone asks that what are the things which we require for our survival, then we say that we need water, that is sure. Then we need carbon means food, organic matter. Then we nutrients, nitrogen and phosphorus. This is needed by all, maybe animal, maybe plant, maybe anyone. Okay now. Micronutrients, many other elements. Then we have energy, for energy, and we know that this energy is produced by oxidation, reduction involving chemical compounds. This is known as respiration. This is, I think this is very basic things. Okay now. Then we know we need electron donor, we need electron acceptor. Okay now, the source of carbon can be of two types. So some of the animals directly get, some organism get organic carbon directly from the external source. These organisms are known as heterotrophics, that is known as animals. Like some animals, yes, some organism directly take external carbon as their food source, and those are known as heterotrophic. Okay, na? But some microorganisms survive on inorganic carbon obtained from external source, which they convert into organic carbon through photosynthesis or other means. These are known as autotrophics. So we are heterotrophic and plants are autotrophic because they convert inorganic carbon to organic carbon with the help of some photosynthesis and all these things. Okay, na? So if we come to know about this heterotrophic and autotrophic organism, microorganism are two types. We already autotrophic and heterotrophic. Autotrophic inorganic carbon as food source, and heterotrophic as organic carbon as food source. Then remember this thing heterotrophy organism use organic carbon as electron donor. So remember how this degradation happens. If you see the chemical reaction here, if you see the chemical reaction here, you can see the here carbon is in +4. Here you can see that carbon is in plus 4, and here carbon is in another form. Means someone is accepting electron and someone is donating electron. So that's why this reaction is happening, and there is a release of energy and all these things. So please mute your mic, I will request. Okay. So remember one thing, that we need this thing electron donor and electron acceptor. So for any biological reaction, if you recall your 12th class, you must have gone through this electron donor and electron acceptor. So whenever we used to write any biological reaction, so there we used to mention that this one is electron donor, this one is electron acceptor, and finally we have this thing, what do we call, bioproduct and all these things. So in for all type of organic matter here also you can see that heterotrophic organism use organic carbon as electron donor. So what will happen, that whatever carbon present in organic matter will donate electron, and they use electronic carbon as electronic sorry electronic I'm sorry organic carbon as food source also. Okay. Then we have photoheterotrophic. Okay, now the those ions which uses light as a energy source. Okay, now however they use organic, say the source of energy is light, but the food source is, means they are using this photon for decomposition and all this thing. Okay, now then we have chemoautotrophic organism uses chemical. So I'm not listing out the examples, but you can always you can take a screenshot and you can see that what are the example of these, because you can see that as a animal which means you can just check, you can just always go on net and you can see the example of each type of organism: heterotrophic, photoheterotrophic, chemoheterotrophic, that they uses chemical compounds other than or other than organic carbon as electron donor, and they use inorganic carbon as carbon source. Okay, now food source. Okay. Then we have photoautotrophic, again same, they we use light as energy source and inorganic carbon as a food source. Then we have aerobic or microorganism which uses oxygen as electron acceptor. Or microorganism itself will accept electron, and they can be either heterotrophic or autotrophic. Okay. Then we have anaerobic organism which uses chemicals other than oxygen. Yeah, remember definition of aerobic and anaerobic. Aerobic is in presence of oxygen, and anaerobic in absence of oxygen. Macron uses chemicals as electron acceptor. They may also be either heterotrophic or autotrophic. So you can just make a combination, that aerobic chemo, aerobic autotroph, aerobic autotroph, anaerobic heterotroph, anaerobic autotroph. And remember, you might have seen gone through this carbon cycle, nitrogen cycle, nitrifying bacteria, denitrifying bacteria, then phosphate, miss many thing. Okay, now so in all those cycles, sulfur miss some bacteria may be oxidizing sulfur, some bacteria may be oxidizing nitrogen. So depending upon, we have different cycle, and in each cycle there is a specific kind of bacteria. Okay, now So I'm not going into that detail that which kind of bacteria is oxidizing which kind of material and all these things. But you can just imagine, like you can like suppose next time if you are reading nitrogen cycle, maybe we have this denitrifying bacteria, just try to identify which kind of bacteria is that. That is that a aerobic bacteria means this aerobic autotroph, yeah, aerobic heterotroph means it cannot be it cannot be autotroph, it will be heterotrophic only. So whether it is a aerobic or heterotroph, yeah, anaerobic heterotroph. So just try to get this idea. So this kind of question are very tricky question. Generally these are not asked, but in I think they are asked in gate examination. I'm not sure. If you are preparing for gate examination, you might have idea. I'm not remembering this, but you can imagine that how thing can be very means if you are imagining how things work, then it will be very very wonderful to you know that we can relate all these thing in different thing. So I will ask you to go through some good book. um huh. So like I don't remember whether it is in Metcalf ED or not, but you can always check it, okay now. So uh, so so I will be going one by one. So till now I have given a brief introduction, brief recap of whatever has been discussed in your lectures, and something where some topics were new also. So so from now onwards I will be going to the question and answer. So I will request you to participate in questioning, okay now. So just in chat box, just write what will be the answer, A, B, C, D. Okay, I will wait for 1 minute, 1 30 second here, 1 minute. So first question is related to what I have discussed few minutes ago. Heterotrophic macroorganism derive energy required for cell synthesis from. So just first remember what is this heterotrophic. I think this is very simple question. Okay, now Divyani Jyot.
C. C. Oxidation of organic matter.
Rupali, are you saying something? Just write in chat box. Okay, na chat box is C. We have got one answer C. Okay, so I will just discuss one by one. So first remember what was heterotrophic organism. Organism that cannot synthesize their own food and instead obtain energy by consuming organic matter. They derive the energy required for cell oxidation of organic matter. The process involves breaking down of organic compounds such as carbohydrate, fats, proteins to obtain energy for growth, reproduction, and maintenance. This is what I have said. So yeah, C is the correct answer. So in photosynthesis is incorrect because photosynthesis process used by autotrophic organism. This thing I have also said, like plants and some bacteria to convert light energy into chemical energy. Then we have this thing. Then we have just 1 minute. Let me make it a small. This cannot be more. Hmm. Hydrogen and sulfur are some chemical. This is also you can see on net. Okay, na. But it's some standard book. External thermo biological process are not source of in biological. We do not need any external source apart from light and all these things. So yes, the C was the correct answer. That is oxidation of organic matter is the correct choice. So like I'm just giving you example like how this oxidation happens. Okay, na. So remember, this is like I have directly taken from net just to explain you that how this oxidation happen. So you can see that one of the most well known reaction of energy generation in heterotrophic organism is oxidation of glucose. So we know that glucose is a very well-known organic compound that is available everywhere. Okay, na. The overall reaction for aerobic respiration can be summarized as this C6H12O6 plus 6O2 plus 6 CO2 plus 6 H2 plus energy is releasing in the form of ATP. So remember, this is not a direct process that if oxygen is available and organic matter are available, so it go through different cycle. Okay, now the different cycle are glycolysis. Okay, now first process is glycolysis. The glucose is broken down into two molecule known as pyruvate, and the step occurs in cytoplasm and generates a small amount of ATP and NADPH. So these are energy pockets. Okay, now ATP and NADH. Okay, now then remember this, we have, you might, if you are aware of this respiration, I think don't miss the of curve cycle. So this is very if you ask from someone from biological department, he or she can explain you in a much detailed way what is this curve cycle. Okay, now so this the compound which was formed is further oxidized into mitochondria, where it is converted into acetyl CO, which is enters the citric acid cycle. The cycle produce additional NADH and FADH along with CO2 as a byproduct. So remember, at each step there is a production of this new what do we call uh, new energy pockets. Okay, now and finally we have this electron accept. Again we have this, there will be some this NADH and FADH which was produced in the curve cycle will combine with O2 and finally produce some ATP, that is energy pocket, a form of energy. Okay, now so the complete oxidation of one molecule, typically it's about 30 to 32 molecules of ATP, which organism use for various molecular processes, including all synthesis. Okay, now so just for your own knowledge, you can just always check on what is this curve cycle. Let me remember. Let me check that whether I have this thing or not. MTech second, MTech first year. Boss, boss. Like I will show you what is the beautiness of this thing. Okay, now how this how miss we are just seeing simple a photosynthesis. Okay, now but in photosynthesis there is something miss in inside this glucose catabolism. You see there is intermediate process which I have said glycolysis, then intermediate process, then this curve cycle, the TCA cycle, then we have oxidative photolysis. You can just check it, electron transfer chain, this, and you can see that this is a whole process how this glucose is simply converted. You can just by simple, if we add all the reaction, we can see that 6 C6H12O6 plus 6 CO2 giving 6 CO2 and all these things. But behind that process a very complex mechanism is always going on. Okay, now so you can see, so I'm not sharing all these things, but just for your if you are in BTech, no need to know this thing, but you can always like just for your curiosity, I'm just showing this just for your to encourage your curiosity, like how the so that you can get an idea how things work. Again, this is our curve cycle. This curve cycle is in part 1, part 2, part 3, and finally this is complete curve cycle. So you can see it has started from a simple acetyl group, and finally it has converted it has given a output as a CO2 and 6 mole of NADPH and all this thing. Okay, now so point of showing this thing that so that you can get a idea how things work. Just don't mug up a thing. Okay, now and where you can find all these materials, you can just always search on right now on internet. Huge amount of material is available, so you can just go and search glycolysis, curve cycle. It will explain you in a very detailed way. Okay. So coming to the second question, in comparison of anaerobic microorganism means which do not require oxygen, or in absence of oxygen. aerobic microorganism have. So you have four options. You have to choose. I request everyone to participate. Very few people are present here, but I request please participate. And as few people are available, it is a good chance to have one-to-one interaction. So if you have any kind of doubt, not related to this thing only, to other topic also, if I am aware of that topic, I will try to answer it. Okay now, so if you have any kind of doubt or anything, you can just ask right now. Only three four people few four people are here, so one to one interaction can happen easily. Okay, so I think I will explain it. So see the definition of aerobic microorganism: use free oxygen as terminal electron accepted in their metabolic means it uses free oxygen as for energy, and which generate more energy ATP per molecule of substrate compared to the anaerobic microorganism. So this the answer of this question lie in the this whole process means how much OTP sorry OTP ATP and all these things are being produced. Okay, now this increased energy availability supports a higher growth rate. However, to sustain this rapid growth, aerobic require so more nutrients, including carbon, nitrogen, and other essential. So just simply, just you can see that this aerobic the answer lie in this option only. That as this aerobic microorganism produce more energy and all these things. So and how we come out to know that it produces more energy? So that is from theory thing. So that I have not explained here how. So if you look the overall reaction, you will see that a high amount of, I will just let me show you if it is in this PDF or not. Aerobic microorganism chemical. No, it's not written here in my. But you can see that this overall. Oh, someone left meeting. Okay. So as I was saying that more energy is produced. So the to support the growth and all this thing, a huge amount of energy is also required and huge amount of nutrition is also required. On the other hand, we have an aerobic microorganism where the oxygen is not available and use other electron acceptor such as nitrate, sulfate, or carbon dioxide. This results in less efficient energy production and consequently lower nutrition requirement due to their slower metabolic rate. Summary means the correct answer is A. Okay. Then we have this thing. I have tried to answer this question. I have explained this thing in detail. You under microbial growth in a batch process, a stationary phase occurs when I have oh no, I think so. Okay, I think you were not present in the session when I was discussing it, but those who were available, please try to answer it. Rupali, do you have any idea of this microbial growth process?
but I'm not sure.
Okay, just for your convenience, I will just repeat it once. So this is like this is our microbial. So what happened that this is our microbial growth, how microbes from birth, what it does, how it passes through different phases and finally it die. So what will happen that suppose this is your system, this is your system containing organic matter, and you introduce some microbes into it. So what will happen that it first the microbes which has been is introduced in this organic matter. So first that microbes will start to adjust itself depending upon the chemical property or different condition type of organic matter. So that phase is known as lag phase. Okay now. So this lag phase is just adjusting, the microbes are adjusting himself, yeah, itself, to the given condition. Okay now. So now once it has adjusted, so what it will see that a huge amount of food is available for that, because organic matters are present now. So that bacteria will started to feel that a huge amount of organic matter is present. So what will happen that these bacteria will start eating those food, and in this way they will release energy, and those energy will be used to generate new biomass, or in simple way we can say that the reproduction of the microbes has increased. So in that way what will happen that their population will increase. So that phase is known as growth phase or exponential growth phase. So as because huge amount of food is available, and a number of as they are consuming those organic matter, so they are producing new energy, and those energy are being used to produce more energy and all, and this go on cycle. But after some time what will happen that as soon as microbes don't have a large lifespan, so what will happen that their cell will start to deteriorate, and at the same time the food which was available has also started to decline. So what will happen that at the rate at which they are producing new cell, at the same rate they will start to die also. And that phase is known as a stationary phase. You can see that the population is almost constant, but and remember at the same time they are dying also, the food is also increasing, decreasing, the amount of food which was there also decreasing. And the as they were dying, they miss they themselves were producing a kind of waste, because where once the microbes which has died, where they will go, they will remain in the system only na. So the amount of food has gone. The main factor is the presence of amount of food. As the amount of food has gone, their production has decreased. So that's why they have come to in the phase of declining growth phase. Remember we are saying declining growth phase means there is some growth, but the rate of growth is lower than the uh, rate of reproduction. So that why it is declining, the rate is declining. Okay na. And then finally the all the food which was there has has finished, the all the cells all the bacteria which was there has almost died. So we have reached into this endogenous phase. Okay na. So the each term I have tried to explain is this is known as log phase. Okay, remember this is lag phase and this is log phase. Okay na. Because the log phase, why? Because this is exponentially increasing. You can mathematically model. Okay now the this is a first order reaction, that the amount of bacteria concentration, that is dc by dt, is proportional to kc. Okay now, and you can model how this, so that's why it is known as log phase. This is a period of intense growth where the population of microbes rapidly increases. How? Why rapidly increases? Because they have abundant nutrients, because they have abundant amount of food. Okay now, during this phase the growth rate is at its maximum as the microbes are actively dividing and replicating in a favorable environment. Okay. Then we have a stationary phase. In this stationary phase, the growth rate stabilizes and the number of viable cells remains relatively constant. This occurs because the rate cell division equal to rate of death. Because availability of food has been reduced. All the most of the 50%, more than 50% of the food has been consumed, and the cells of the microbes are dying. So rate of division or cell rate of reproduction is equal to the rate of death. It is typically characterized by plateau in the growth curve, nutrient depletion, waste accumulation, and other environmental factors contribute to the exponential growth and the entry into the stationary phase. So this thing I have already said. Then we have declining growth phase. After exponential growth phase, the growth rate begins to slow down due to depletion of nutrients, accumulation of waste products, and the other factors. In this phase, the growth rate decreases and the number of viable cells may start to decline. However, it is important to note that not all cells may be dying at this stage, but overall rate is very small. I hope this is clear to you. Then we have this indigenous phase. This is phase where the number of viable cells start to decline. The number of and the cell enters a state of dormancy or programmed cell death. This phase is characterized by a decrease in cell viability due to various factors such as uh, nutrient exhaustion, toxic metabolic accumulation, because all the waste, all the macros have died, so a toxic condition has occurred, and the environmental stress. Eventually the population may reach at a point where most of cells are no longer viable, leading to the decline of culture. Okay, so this was our microbial growth phase. Okay now, so coming back to the question, try to answer it. A stationary phase. So you can see this was our stationary phase. This was adjusting period. A huge amount of food, rate and death are equal, and here there is only death. So rate of growth nearly equals to rate of, the answer will be B. Rate of growth become nearly equal to the rate of decay. If you have any kind of doubt, you can ask. I can explain. Please don't hesitate to ask anything. Already we are few in numbers. Any doubt? No, then I will be going to the next question. Question number 4. So this thing is like so again I think you were not available during this discussion. So this is a picture of our ideal activated sludge process. There is this tank, then there is the activated tank, and from activated tank we are recirculating this some microbes to activated microbes to decompose our organic matter. So where X is biomass concentration and S is substrate concentration. Okay now. So depending upon how this X is varying and how this S is varying, we have different formula and all these things. So that based on that we define different terms like what, at which rate there will be degradation, at which rate the organic matter will decompose, at which rate microbes will increase and decrease and all these things. Okay now. Huh. So I remember already answer is marked, that is B, but again I will be going. If S donates BOD, X donates MLVSS in form mixed liquor, volatile suspended solid. In many book, in many places you will be written, it will be written as MLVSS. Okay na, and T is the time, match the following expression. Okay na. So first we have dx by dt. So dx by dt represents the the rate at the change of biomass concentration over time, which is commonly known as biomass growth rate. Okay now. Then we have dx by ds. This expression represents the rate of change of biomass concentration related to the change of substrate concentration, that is BOD. This is known as yield coefficient. Okay now. Means like for particular amount of biomass degradation, how much substrate is getting consumed. So this is kind of a specific coefficient which is given for. We will be looking what is the use of this dx by ds, because it indicates how much biomass is produced or consumed per unit of substrate consumption. Okay now. So remember, at one point there will be production of biomass, at other point there will be decrease in biomass. So you can just assume, you can just have an idea what is the ds by dt. Then finally we have ds by dt. This expression represents the rate of change of substrate concentration over time. It is commonly referred as rate of substrate utilization. So here you can see that this will be matching with B, this will be matching with C, this will be matching with A, and the correct answer will be this thing, that is B. B will be our correct option. Okay now. So remember this thing. So if I start to explain what its different term, it will be very, I mean, not difficult, but it will be very time consuming. Here I am just to recall your whatever you have studied. So just remember, try to have a control whenever you are studying this activated sludge process. Try to have a control on different terms: dsy dt, dxy dt, dxy ds. So in this way, you won't be getting any kind of problem in solving numerical question. Okay, na? So we have some numerical question at the end of the lecture, and we will be seeing how these are being used to solve different kind of question. Okay. Then we have this thing biological. Remember this biological treatment of wastewater. We are just focusing on biological treatment. So in this biological treatment, what we are doing, we are degrading organic matter or we are removing organic matter. So in removing organic matter, what are we removing? We have four options: POD, COD, yeah, only BOD, both BOD, COD, or neither BOD, COD. So please try to answer it, that if we are removing organic matter, what we are reducing in our system. So remember, suppose if you are working in wastewater treatment unit. So now what happened that you A is option, we got A, okay. So just try to see the physical, means real world implication of this. Why we are, a few minutes ago we were discussing organic matter, and now we have come to BOD and COD. Remember, COD is chemical oxygen demand, and BOD is biological oxygen, biochemical oxygen demand, not biological. Remember, biochemical oxygen demand. Okay, now. So remember now what will happen, that suppose if you are working in wastewater treatment unit, and now you won't be measuring organic matter, we measure COD and BOD of our system. Okay, now. So there is one general procedure that how DO and that is dissolved oxygen is measured, and depending upon the dissolved oxygen, we can calculate this BOD and COD. And remember, this BOD COD is chemical. So I will be going one by one option. The correct answer is both, that is BOD and COD. Remember, COD is combination of BOD and first try to know what is this COD. COD is chemical oxygen demand, which is means whatever kind of chemicals, maybe organic, inorganic, whatever chemical is there, it will it the oxygen which is being demanded by that thing is known as chemical oxygen demand. And we have this biological oxygen demand, which looks to this organic matter. So remember, this BOD is always greater than or equal to COD. Sorry, COD is always greater than or equal to BOD, where BOD is on the top, combination of organic, inorganic, both. But when we are looking at a BOD, it always focus on just name suggests that it is talking about biochemical. Biochemical means organic matter. Okay. So the involves process where where microorganism you sorry where microorganisms are used to break down organic matter in wastewater. This treatment is effective in reducing both biological oxygen demand and chemical oxygen demand. So I will be going to this BOD and COD. BOD, which stand for biochemical oxygen demand, measure the amount of oxygen required by microorganism to decompose organic matter in wastewater over a specified period, usually 5 days. Okay, na. Since biological treatment process target organic matter, they significantly reduce BOD level by utilizing microorganism to consume and break down the organic pollutants. Okay. The COD measures the total amount of oxygen required to oxidize both organic and inorganic substance in the wastewater using a chemical oxidant, using usually potassium dichromate. Biological treatment can also reduce COD because they reduce significant portion of organic compounds that contribute to COD. Okay, most of the demand which of oxygen is due to carbon only. Okay, now all organic matter only. So that's come under COD also. Okay, now so COD on the top. On that below we have BOD. Biological treatment process such as activated sludge trickling filters can reduce both COD and BOD as they work to remove organic matter and reduce overall pollution level in the wastewater. So the correct answer will be C, both BOD and COD. Okay. Then we have another things which of the following is not a component of typically activated sludge process. We have secondary settling tank, aeration tank, biomass, silica media, and then sludge recycling. So just from definition, you can see we have discussed all these things, all, and there is one topic which we have not discussed here. Okay, now. So what does this activated sludge process does? It break down organic matter or reduce BOD, COD, and all these things. So what will be there? It will degrade organic matter which is there in our wastewater, and once degraded, it will be start. This is again, I think, biomass growth media. So how we in our system we have this biomass growth thing, na? Microbes are being introduced, organic matter is there, so there will be a growth of biomass. So this is our simple a ASP which we have discussed. Okay, now. Huh. So remember, biomass growth media. Huh. So yes, correct answer is C. But just remember where this here we have this suspended thing. Okay, now in ASP process we have suspended. We don't have any kind of media. Media. Can you say in which media? Where we have media? We have media in this trickling filter and RBC, that is rotating biological. So I think this was a simple question. Coming to the another question, simple question. Activated sludge process based on the include treatment that are based on attached growth. Remember attached growth. This ASP is our suspended. So this won't be our answer. This won't be our answer. This won't be our answer. Okay, now. So the correct answer is obviously A. B, but we should know what are these things. Attached growth system are wastewater treatment system where microorganisms grow on surface or media, allowing the biomass to attach to the media rather than suspended in the media. They the key system on the include trickling filter. We have trickling filter. The system uses a rotating bed. I have so rotating bed such as plastic or stone. No, not rotating bed. There is no rotating. Simple bed is there. I think I have written it incorrectly. Okay, now. So let me show you the picture of this trickling filter. I think I don't have pictures right now. I think there was one picture. Just 1 minute. So see, this is my trickling filter. Okay, now. So it will be organic. So wastewater will be coming there. So you can see, you are able to see my screen now. Ah, so you can see there will be a chamber. There will be some media, and media will be like this kind of thing. Either some kind of a special type of plastic and all this thing, or a porous, this type of structure which will be laid all over this tank, okay na, and wastewater will be sprayed from the top, okay na. So what will happen that with time you can see suppose imagine that this was one media, here there is organic matter, sorry, here there will be a biomass, the water will be coming. Trickling means I think you can imagine what is trickling. Trickling means water is passing on the surface of the uh, uh, when water passes to any surface we say that it water is trickling. This, I think, I don't know. I'm saying correctly only. So water will trickle over this biomass, and in that process, there will be this degradation of organic matter. Okay, now. So this is a picture of trickling filter. So here this one is rotating, but there is no rotating. I think I have written it incorrectly here. Rotate, not rotating bed of media. Use a bed of media. Only bed of media is not rotating. Okay, na plastic over a wastewater flow. Over is wastewater flow. Huh? The microorganisms grow on the media, forming a biofilm. As the water moves over the media, microorganisms degrade the organic pollutant. Okay. Then we have RBC. This system consists of rotating disc or media that are partially submerged in wastewater. Microorganisms grow on the surface of the rotating. Then this rotate. The biofilm and the media comes in the contact with the wastewater, allowing for the treatment of the organic matter. SP is suspended, so the sensor SP we have already know that it keeps in suspension in aeration tank, so it does not rely on the attached growth hub. And this is the picture of this RBC. You can see these large plates. I hope these are clear to you. Question number 7. Huh. So, uh, so we have this thing. Now this is related to some what do we call mm, as what do I call? Huh? Some formula related to ASP. Okay, now then we have sludge volume index, organic loading rate, solid retention time, food to microorganism ratio or F by M ratio. Okay, now. So remember the question in activated sludge process kg of BOD applied means whatever the BOD that is biochemical oxygen mine applied per unit ml VSS. Told you na this mixed liquor, volatile, suspended solid is sometimes referred as ml, and this is very common term. Everywhere it is used. Okay, na is referred as. So we'll be looking. So remember we have different terms related to this. If you just go to any book, yeah, just search on net list, just search list or all the formula related to activated sludge process. They will list out to you every formula, what is loading, how much oxygen, how much microbes, what will be the after treatment, what will be, how can I determine what will be the BOD and COD of I think you are getting it. So see, here we have different terms. Q that is flow, S note we have, X note we have, C note we have, initial concentration, initial biomass, initial subtract. After treatment we have this thing. So there is a relation between each and every time. There is relation between X note, XR, XC, X XR, then the C note, CR, there is then CE, there is a relation between relating all these terms. Okay now, and different terms has been defined. So one of the most common terms which is there, we have hydraulic retention time means how much time this waste will be here in this tank. So that is simply V by Q naught, that is volume of the tank, and Q naught. So this helps in design of our biological reactor. Okay, then we have subtract utilization rate. At what rate the subtract, that is S, or organic matter is getting utilized. So that is known as Q naught, how much it is getting. Initial S naught which was coming, and after treatment we have S, means there has been some conjunction of abstract into divided by volume into X naught, depending upon the miss volume is the volume is v the volume of the tank, and x is our biomass. Okay, just let me check. X is our biomass now. X is our biomass. Biomass means this organic matter. Okay na. Where does it go? Huh. So this is known as subtract utilization rates. See, you can just simply imagine, and you don't have to mug up those formula. And then something we have food by mass ratio means how much food is there, means how much organic matter is there, and how much how much biomass is there. That is food by mass ratio. How much food is there, how much microbes are there. So that is known as FM. So generally a question is asked that if it is a ratio, what ratio generally, if you have seen anywhere, ratio is unitless quantity. But can you say what will be the unit of this F by M? Anyone in the audience want to answer it? What will be the unit of food by mass ratio? Here the HRT unit is time. V meter cube. It will be meter cube per day, and meter will get cancelled out. We have meter cube, so it has a unit of time. Similarly, there will be some unit of this thing. It is milligram per liter. It is also milligram per liter. So milligram gone. Then V is the volume. Then utilization rate rate means time also. So it has time minus one. Okay, na T minus one. Similarly, anyone just put the unit and just try to find out what will be the unit of food by mass ratio. Take your time, take your pen and pencil, just try to answer it. So see, S note and X will be in milligram per liter. So milligram per liter cancel out. This Q will be in volume per time, and will be in volume. So volume will get cancel out, and you will have this one one by time. Okay na. One by time. So its unit will be also time inverse one. So unit of this utilization rate food by mass ratio will be minus one. Okay na. Then we have something called I will be explaining food by mass ratio. In food by mass ratio is the amount of BOD, that is biochemical oxygen demand, applied to the ration tank per unit mass of mixed liquor, volatile, suspended, solid. It is critical parameter in the design and operation of activated sludge system, as it indicates the balance between the organic food entering in the system and the microbial population available at to treat it. So see, we should have always idea that in our activated sludge process, what will the population of microbial, and depending upon the population of microbes, we should add that much of organic matter. Okay na, it's not like that or at equally works in different way. Like like if the organic content is very high, we add high amount of microbes in our system. If organic content is very less, we add less amount of uh, we add very less amount, because what will happen that suppose if you add too much food means what will happen, um, just imagine too much food is there, so you have to too much food for microbes is there, so what will have happen that uh, remember one case that a huge amount of organic matter is very high but the microbes is very less. So what will happen that obviously all the organic matter will not get decomposed, so you have to add more, so it will be waste of resource. Now what happened that a very less amount of organic matter is there, and the microbes is very high. So what will happen that all the macros will start to die because they have very less amount of food. Okay na, so we have to maintain a balance, and we have to find a optimum value of microbes depending upon the amount of organic matter. So that's why this food by mass ratio is very essential. Then we have sludge volume index, measures the settling characteristics of sludge and is not directly related to amount. This is just simple. You might be, if you are you might be doing this kind of experiment in your btech also, that a wastewater will be given to you, and a conical flask, this SBI, this type of conical shape of uh, flask is there, and wastewater is added to it, and then it is allowed to settle, so it looks like this. So this thing, so this picture is, I'm not remembering the name of, but this is used to calculate the SVI, that is sludge volume index, means for 1 liter of volume, one generally it has capacity of 1 liter, generally it is 1 liter only, huh? 1 liter of waste is added, and how much sludge has settled at the bottom of this tank is measured, so and it is reported as, suppose the volume, and it is marked in ml. So suppose after 1 generally it is kept for 1 hour, 1 hour or 24 hours, that I have to see, and also it's d. Okay na, so generally, mm, generally for 1 hour it is kept, I'm not remembering, 1 hour or 24 hour, and suppose it is 10 ml, so we say that it is 10 ml per 1000 ml. Okay na, so this is how SBI is defined. Then we have organic loading rate. It typically amount of organic matter applied per volume, how much organic matter is being applied. Then we have SRT, solid retention, the refer to the average time that biomass remains in the system, and it is not same as food by mass ratio. Remember, okay? So the correct answer for this question will be uh, food by mass ratio, D. Yeah, you were correct. So remember, before going into, I just want to show that what are the different formula which is generally used. So these are direct from my lectures, so that's why I have not added in my slide. Okay, now because this will be a copyright thing. So remember, I have already said just try take a screenshot, and you can just, and these are also available in book also. Okay now, so just either you can just have a look, huh. So suppose this is x is our biomass concentration, ac subtract means ac is our organic matter, and x is our microbial. Then we have already seen that ds by dt is known as subtract utilization rate. Then we have substrate utilization rate per biomass concentration, that is known as a specific substrate utilization rate. Then we have ds by dt total, that is known as gross biomass growth rate. Then we have g, ds by dt g, that is known as net biomass. Like there, see, these are different terms which is and after some time we will be seeing the relationship between these terms. Okay now, then we have ds by dt g per x, that is net per biomass. So that is not a specific net biomass growth rate. Then we have dx by e, that is biomass decay rate. That x means biomass at what rate this is decaying. Then we have dx by dt e per x, that is a specific biomass decay. That for particular biomass concentration, how much biomass is degrading, that is known as kd. Okay now. Then if you look at look at the relationship between each term, so we know that this gross will be net, sorry, this net will be total plus this e, that is by dx by dt. So see, dx by dt g, it will be dx by dt t plus dx by dt e, and similarly dx by dt t. So you can just substitute different terms, and finally you will be getting this relationship, that mu, that is a specific net biomass growth rate, is equal to by t. And what was our by t? By t was a specific yield coefficient into q. What is q? Q is a specific utilization rate minus kd. What was the kd? Kd was a specific biomass. So this is a very useful relationship. Okay now. Again, this ds by dt is equal to minus by observed by, so this is theoretical thing. So this is what is observed. So depending upon it has been observed, it has been seen that dx by dt gross, that is that is net biomass is equal to absorbed into ds by dt. And similarly you can substitute here, and you will be getting a relationship that mu, that is a specific net biomass, is equal to observed yield coefficient into q. And you can just rewrite it, you can just substitute the u, substitute u from mu from here, and you can find the relationship between by theoretical, yeah, by t, that is yield coefficient, yeah, and e yield observed into minus kd by q. Okay now. So again, I was saying that for each terms we can mathematically, this is known as lag phase. Means system is adjusting itself. This x is low. What is x? X was, what is x? X is our biomass. Means biomass is low and s is high. So mu will be high. You can just make a table that which will be high, which will be low, and all this thing. Okay. So this is representing our food, and this is representing our this thing biomass. So you can see that initially food was high, and after treatment it is very low. At the same time population is increasing and all this thing. So x versus s, it has been plotted, and different what time, where x will be high, x will be low, x will be declining, s will be zero, s will be high, s will be sufficient, s will be high. So this is, just take a screenshot of this thing also. So remember, there is one famous kinetics. So we know that first order, second order, third order, but generally this that system won't work here. So we have a different type of kinetics, that is monode kinetics. So it is said that when q is half of the q max, then we have this uh, first order, sorry, I think you can see yes, when s is large, s is very large, s is very large, then if s is very large, then we will have q is equal to q max. And we know that q is, what is q? Q is equal to ds by dt. So for this thing we are zero order law, zero, initially it is following zero law. And after some time, when a case will be when for the case where case will be high, then this can be neglected, and then we have dx by dt is equal to q max by divided by kh into s. Then it is following first order reaction. So you can see that initially it was means depending upon the condition, it will be either first order reaction, zero order reaction, and first order reaction. Okay now, just again this is some formula. So remember, we can derive many formula based on this mass balance equation. We have seen, if you have attended my second lecture, I have given you some book chapra book, Steven C chapra. By taking that reference, you can derive different formula and all this thing. Okay now, again I am not going into this. So just take a screenshot of this question. Answer is already given to you 4.1. I forget to remove it. And the formula for u mu is equal to mu x into s divided by ks. So this specific, this we have defined right now in monode kinetics. Okay na, that is mu is a specific growth rate. Here I have written in the term of q. Okay na, see, different book uses different unit. It's all, but it's all same. Okay na, here we can see q max is equal to q is equal to q max, but in my solution I have written q mu. Okay na, so mu is equal to mu max s by ks by ks. And in your question everything is given to you. Just try to answer it, this thing and this thing. First I have just write this formula. Once you have written the formula, I hope you have written it. This is the question. Okay now just try to solve it. I'm coming in one. Try to solve it. Answer is 4.1, but you have to like identify each and every time. Not, you don't have to identify it, it's already given. A specific growth rate mu was observed at itna bod is given to you. Mu is also given to you. The case, that is the half saturation index is of a species is 48. So you have to apply one thing, that then we have to find mu max. So here mu is given, s is given, ks is given, and you have to find mu max. So we know that the formula is mu is equal to mu max is equal to s divided by ks plus s. So we will be just rearranging it. We will be putting all the value, and after solving it we will be getting the mu max is equal to 4.1 per day. Just to remember, this unit is of this mu s is per day. Okay now, then we have second question. We have a tank of 4500 meter cube size. A recent tank of an activated sludge process is maintaining steady biomass concentration. See again, biomass concentration. Just you have to see. I can feel. Miss, I can say that the in examination it will be difficult for you to memorize the formula. So, but the one thing you can do, that on the spot you can derive all the equation, but it will be very time consuming. Other option is just to mug up all the formula. But before mugging up, you should make sure that you are understanding each and everything. Okay, na? Or I won't suggest to mug up the formula, but I think you are. I think you should somehow either you just drive it on the spot, or somehow just use your basic concept of how this every term is related to each other, and you can solve the problem. Okay, now biomass concentration is given to you. Q is given to you. And then we have biomass concentration is the Q, while the biomass concentration is 0, the biomass which is coming out to be has given to you, then approximate sludge retention time. So the important question is, first you should know what is the formula of sludge retention time. Okay na. So this sludge retention time formula can be again by, if you, so I have used this graph, miss. So all the first you should know what is sludge retention time, what is the definition of sludge retention time. I think I have written it here. Sludge detention time refers to the average time that biomass remain in the system, and we know that this is our system. So depending upon this, we can just. I'm not going to derive it, but you can just imagine it. First, you have to find out where your system, where your where your this biomass will be residing, it will be residing here, and what are the different thing which is coming here. Okay na. So depending upon it, you can just find out the expression for SRT, and SRT will be this thing. So that is V into X, that is volume of the tank, into biomass concentration divided by QW. QW, what is the flow in the, we have seen that na ki, uh, sorry, in our tank, this something is coming, the wastewater is coming, so QW is the sludge wastage. Then we have biomass concentration is the sludge wastage. Then Q is the effluent flow rate, and X is the biomass concentration in the effluent. Okay na. And everything has been given to you, so the most important thing is to you should know this formula. Okay na. So if V is given to you, X is given to you, QW is given to you, QW is given to you, XW is given to you, Q is given to you, X is negligible, so that's why XC in this formula, XC and QE all will be 0. So your final expression will be Vx by V into X divided by QW into XW. So you will be substituting all the value here, and finally you will be getting SRT at 5 day. Okay. The question was very easy. Only thing which you have to do was to just to know this formula, SRT formula. If you have any doubt, you can just ask it right now, or I will go. We have more three question. So the first is again all the question is related to activated sludge process only. An activated sludge process to treat 6.4 mld sewage having 320 milligram BOD at the volumetric organic OLR is given to you. Okay. The design so I said you organic loading rate is like how much organic is coming to at the what rate and all these things. The design steady state BOD and the MLVSS mixed liquor, volatile suspended solid concentration in aeration tank are 20 milligram, 20 milligram per liter and 2000 milligram per respectively. The oxygen requirement for the process is estimated as 1.72 oxygen per kg decay removal of VOD. Based on the information, we have three questions, and I will be going one by one. So the effective volume of aeration tank should be. So I will be giving you 2 to 3 minutes. Just try to solve it. Just use this figure. Use this figure. Take a screenshot right now. Just try to draw it. Just see what information has been given to you, and try to, based on the information given to you, try to find out the volume of this. We have to find the V. Okay now. Take your time. Take your time and try to solve it. The effective volume of aeration tank should be in meter cube. So this it's a simple question, very simple question. Like it's saying that there is one. Suppose I am not drawing everything. So a wastewater is coming that has a capacity of 6.4 million liter per day and has a BOD concentration of 320 milligram per liter. So it has to be treated. It has to be treated. Okay na. So So you can see that first you should know, I have already said that what is volumetric organic loading rate is Q into S naught. Q is how much flow is coming, S naught is influent BOD concentration, and is the volume of the tank. Okay na. So this is how OLR just you can just remember, first thing you should know that if something is being talked about this rate, so it will be something per time. Okay na. So now as it is talking about organic loading rate, so at what rate the organic matter are coming. So if we are talking about organic thing, so what does for our question we said that we won't be measuring our organic matter and all this thing, we will be measuring BOD. So at what rate this BOD is coming. Okay na. So we know the incoming thing, that is at this rate the flow is at this rate, and it has a unit of meter cube per time. So see, if you have everything cleared in your mind, so you don't have to apply this thing. You can just simply say that at whatever rate this organic matter is coming per unit volume will be our organic loading rate. But sometime if you are confused, just try to match the unit. So it is talking about organic loading rate. Organic unit is milligram per liter, and it will as it is of rate, so it will be milligram per liter per time. Okay na. So so that's how you can also apply, you can find the formula, but you don't have to. So this is like a jugaru kam, like we are just somehow arranging things. This is not a good concept, but just to make you understand how things work, I'm just saying you all these things. Okay na. So Q into S naught, in your question we have already given this Q is equal to 6.4 mld, and we can always convert mld means million liter, and million liter means 10 ki power 10 की power 6 liter, and 10 की power 3 10 की power 3 liter is equal to 1 meter cube. So we can just I have multiplied 1000 with 6.4, then that is 6400 meter cube per day. S naught is this thing 320 milligram per liter, and we have converted into milligram to kg and liter to meter cube. Kg it is 10 की power minus 6 10 की power minus 6 kg. Meter cube is 10 की power minus 3. So putting all the value, it is 3.2, and organic loading rate already it is given to you. So 0.5 kg per meter cube per day. So I will be rearranging the formula, I will be substituting all the value, that is V is equal to 60. So we can say that V will be Q into SL naught divided by OLR, and already we have given to you this. So see, you can see this m meter cube will get cancelled out, then this day will get cancelled out, this kg will get cancelled out. No, this meter cube, not. Remember which meter cube will get cancelled out. 1 meter cube will be saved. So this and this will get so this and this will get cancelled out. Eventually, this will go up. Okay, now up. So finally, you will have this thing that V is equal to 4696 meter cube. I hope this is clear to you. Then food to mass ratio. Try to solve it. I have already shared the formula of this question also. You have to calculate F by M. Just remember what was food by mass ratio. What was food by mass ratio? How I have defined food by mass ratio is how much organic matter is present. Sorry, how much food is present and how much mass. And what was food? What was mass? Food was when we are talking about food, we are talking about organic matter, and when we are talking about mass, we are talking about the mass of this microbes. And in this question, everything is given to you, MLVSAs and all this thing. For your convenience, I am just showing the formula of food by mass ratio. Food by mass ratio is given by Q naught by S naught divided by V into X. You just right now we have calculated V. X Q naught just note down this formula. Use this whatever the information has been given to you, and try to find out the answer. What will be the food by mass ratio? this was so see, I have already said that to calculate the food by we use the following formula: food by mass ratio equal to Q into S naught. So food means see S naught means BOD. Food, what was our organic matter, and how organic matter is quantified in activated sludge process? It is quantified by the means of BOD. Only way to measure amount of organic matter is in the form of BOD. We cannot measure directly how much organic matter is there. Only way to quantify the amount of organic matter is BOD. So so this is very general practice, BOD, COD. So we have measured BOD, and we have a volume of the aeration tank, and X is the biomass concentration in the aeration tank, or MLVSS in milligram per liter, or kg per meter cube. So I think everything is given to in this question. MLVSS is given 20 milligram per liter is given to you, and sorry, MLVSS is 2000. This is design BOD. Can anyone say what is this design BOD? What is this? What do you mean by design BOD? It's just simply saying that whatever this COD is coming, BOD is coming, that it is coming 320. After treatment, it should be 20 milligram per liter. So this has been designed in a such a way that if sufficient time, retention time, and all this thing is being provided, then what will happen that the wastewater coming after from this secondary tank will have a BOD of 20 milligram per liter. Okay now. So here everything is given to you. Q is equal to 66.4, that is 64 meter cube per day. This S note is 3320 milligram per 0.32 kg per meter cube. V barabar this thing we have calculated, X we have 2 kg. So I will substituting everything, and you can say this kg will get cancelled out, and the final you can just report your answer in 0.25 per day. Okay now, no need to write this much, because I have already discussed this. The unit of this food by mass ratio is day time inverse. Okay, that is day minus. So although it is ratio, but it is not a unitless quantity. Okay, I hope this is clear to you. Coming to the last question, the daily oxygen demand. Okay now, daily requirement of oxygen in kg per day. So to calculate daily oxygen requirement for activated cells, we need to consider the amount of BOD removed. Okay, so remember amount of BOD removed, and amount of BOD removed, they have already said that 320 is coming, 320 is coming, and in question, so if you read the question, it all the data has been given to you. The oxygen requirement is estimated as 1.72 kg oxygen per kg decay, means whatever the amount of COD is being removed, of that 1.2, and this you can, I don't remember it is exactly 1.7, sorry 1.72, but you can just by writing the formula, miswriting the all the chemical reaction, you can just derive that how much oxygen is required for the degradation of 1 mole of organic matter. I will just at the end of lecture I will just see. Okay now. Hmm. So you can see that it is BOD remove into biological oxygen requirement per unit BOD. BOD remove is given by Q into S naught minus S. Q is the flow of sewage. S naught is the influent, that is 320. S is the effluent, that is milligram per, that is this is our 320. This is 20, and oxygen requirement per unit BOD is given 1.72 kg oxygen per kg BOD. So finally the our formula will be, so I believe substituting all the form, that is Q is equal to this, S naught is equal to this, S is equal to this, oxygen is 1.72, BOD is removed into Q into S naught minus S. So BOD removed into 6,400 meter cube per day into 0.32 minus 0.02. It will be BOD removed is equal to 172 kg per day. and we know that oxygen requirement is 1.72 of whatever is being removed. So 1920 into 1.72, and it is coming out to be 3302.2 kg per day. So this much oxygen you have to pass. So if this was your aeration tank, this was our aeration tank. So in this aeration tank, this much of oxygen has to be provided for proper functioning of our for proper functioning of um, our activated sludge process. If it's not that the case, our activated sludge process won't work. And the first thing which we will get, that our demand, that our targeted BOD, that was 20 milligram per liter, won't be achieved. Okay. So 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 in this way, so see now how things work, how when we are designing activated sludge process, there are some specification that our targeted BOD should be this much, our retention time should be this much. So depending upon the retention time, organic loading rate, we just try to find out. Just I will show you, miss. I don't remember, miss. I will just show you a design of activated sludge process. Activated sludge. So you can see that here we have the bold one are the our the in this question, in this PDF, whatever is the bold thing. In bold there is two things now. One is bold, this is 50 is bold. So this is our like this is known things for design purpose. This is known thing, that is flow. That is, suppose if I have a, if I am uh, making a treatment plan, then we will be knowing that how much water is coming. So this Q is knowing. Similarly, this BOD will be also known to us, that how much BOD, how, what kind of organic, what kind of wastewater is coming to our treatment. So this will be also known. Hydrogen retention time we can just define with for how much we want to treat. So depending upon that, we can find this volume of the aeration tank. Depending upon the budget and all these things, we can find how much how many tanks we can provide. So you can see that once we know the number of tank, then we can define volume, then we can define surface area and all these things. Okay now. So these things are all reverse engineers, that one thing. So now imagine, now see this thing, this effluent BOD means this is our targeted BOD, that I have to target this much BOD. Okay now. So depending upon, and this case is all theoretical cheese, QM is all theoretical cheese. Then depending upon this thing, Q and X and all this thing, we can define. Okay now. So this is like a reverse engineer thing, that how much if we know some specific thing like this much is known to us, then we can always design something. I'm not going into detail, but I have just given you a gist, like overall idea how things work. Similarly, this is in more detailed way. Here we are defining each and everything, how much oxygen should be provided, how much area should be provided, how much. So in real world, if you ever get a chance to visit a wastewater treatment, just go, and whoever the engineer is there, just ask the what are the different design parameters. You will just amazed to know that how they have worked out to, like, just to make a that treatment unit that will treat our water and all this thing. Okay now. So this was all for today's lecture. We have 5 to 10 minutes. So if you want to ask anything, you can ask. It's like whatever I was supposed to discuss, I have already discussed. Still, if you have any kind of doubt, please feel free to unmute yourself and start, or you can just write in chat box. It's good that very less people have joined, so you can just have we can have just one to one interaction. Rupali, doctor is S. Ramya. Then we have Am I Bad has left the meeting. If anyone have any kind of question, he or she can ask. There are two one to just go through these book for more detail. That is Metcalf and go through this book for more. This book contains they have discussed about this ASP, that is activated sludge process, and all this thing in a great detail. So I request you to, if you are not able to ask right now, just just you can go to the book. Then when I was talking about this thing, kinetics and all this thing, just go to this book. Where does it? Mass balance and all. I was saying about mass balance. Now just go to this book, modeling. Chapra, 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 chapra. Surface water quality modeling. This is the best book known for modeling thing. If you go on internet and search that, please suggest a book for water modeling. They will suggest this book only. So here also there are many things. Not that I mean, you can just have a basics from this book, and you can always interpret
ये
from
अब
this book.
आ जाए बगल में छोड़ा है भाई
You can mute yourself, leader.
हवाई नहीं आती नहीं छोटे छोरा ही होके छोड़ा है
Mute yourself, please. Huh. So actually we were ending the lecture. So if you have any kind of doubt, we have 4 minutes, 4 to 5 minutes. You can ask. The recording of this session will be uploaded on the this YouTube channel. I think the link for which has been already shared with you, but still I am sharing it. Any doubt, any question not related to this? If you have any other doubt also related to subject material, you can ask. Or else we can end our session here and we can meet next week. From other two members, Rupali, if you have any question, do you have any question? Thank you. What about Doctor? Sorry, Doctor S Ramya. Do you have any question? No, sir. Okay, then I am ending this session. Please join next week also, and next week also. Thank you. Good night.